was the same question in my mind. How was antisense administered, oral or intravenously, and what dose? Yeah, I also was curious about the route of treatment.
The drug, the antisense were administered intravenous. Now we have two different approaches to deliver the drug. One is intratumoral, which is for the glioblastoma. That we directly administered the drug through a catheter to the tumor. And you don't need a whole lot of drug because you're essentially putting the drug right where it's supposed to be. Now, if you're dealing with tumor that easily accessible by intravenous route, then that's how we're going to deal with it. The pancreatic cancer, melanoma, colorectal, those we deliver the drug intravenously. So the dose is higher. Our safe dose was roughly about 300 milligram per meter square, but our effective dose was 140 milligram per meter square. So we chose 140 milligram per meter square, given the fact that we could never hit a toxic dose, or we could never see toxicity as far as we go in terms of the dose. So yeah, we chose the lower dose because it's already showing you efficacy, but I don't think there's an upper limit.
Okay, thank you very much for addressing the last question. And as I mentioned now, I would like to introduce our next valuable speaker. Now, Professor Pan will present. Professor Pan has long studied the molecular and cellular mechanism of opioid actions that are mediated through mu opioid receptor splice variant using multidisciplinary approaches. These studies provide the foundation for developing novel analgesic drugs to treat pain devoid of many side effects associated with traditional opioids. Dr. Pan is a co-scientific founder at Sparian Biosciences, a company devoted to the development of safer alternatives of opioid drugs. And today, the title of his presentation is The Holy Grail of Safer Opioid Targeting Truncated 6 Transmembrane Mu Opioid Receptor Splice Variants. So please welcome Professor Pan, and we are ready to hear and watch your presentation.
Hello, everyone. So can you hear me?
Yes, we can hear, and we can also see your presentation. So let's continue.
So first of all, I'd like to thank the organizer for allowing me to present here today. My lab have long studied the mechanism of mu-opioid actions. Today, I'd like to talk about one of our stories, how to translate our basic science into development of safer opiates. This was one of the earliest anti-pain medicine made by Dr. Bells in 1895. The main components were the extract from opium, contain morphine, heroin, and other opiates. In 1898, Baylor marketed heroin as a cough suppressant and a non-addictive substitute for morphine. At that time, they didn't know the heroin has more abuse potential than morphine. It was short-lived and off the market in 19. This slide shows the drug overdose deaths in US in past from 1999 to 2017. In 2017, is have over 70,000 overdose deaths for all the drug. However, 2/3 of them contributed by the opiate overdose. We have recent data indicating in 2020, the opiate overdose rate is over 90,000, almost double compared to 2017. In 2017, about 11 million people misused the opiates. About 20% of them have opiate use disorder. The main reason for the opiate misuse is pain, over 62%. So what is pain? Pain is subjective sensation or perception induced by non-perceptive stimuli. What the opiate does is to activate endogenous pain modulatory system, mainly through the opiate receptors. Unlike a general anesthesia reagent such as isoflurane, opiate relief pain without altering level of consciousness or impairing overall sensation. Pain is still there, but just not felt. You can still hear this whistle. Opiates are still the mainstream of the pain treatment, mainly for moderate and severe pain. And the opiates are mostly effective in acute pain, cancer pain, post-operative pain, but less effective in neuropathic pain, as well as inflammation pain. I didn't list it here. Developing opiate drug was far lagging behind the other type of drugs, mainly because of potential side effect risk. You all knew the Purdue Pharma who filed bankruptcy in 2019 due to overwhelming lawsuit of oxycodone. However, the pain markets offer the great opportunities. The early pharmacology studies defined the three types of opioid receptor, mu, kappa, and delta. Molecular cloning lead to identify their genes, proteins, MAB, CAP, DOP, and NAB. And they all belong to GPCR class A rhodopsin family. There's a fourth member, NAB, structured very close to other three opioid receptor. However, this knob doesn't bind any opiate. The crystal structure of all these four members of receptors have been resolved and published in the same Nature in 2012. And the mu opioid receptor have a special place within the opioid receptor family because mainly it mediates the action of most clinically used opiates. Such as morphine, fentanyl, methadone, as well as drug abuse, heroin. This is a crystal structure of mouse mu opioid receptor. Is co-crystallized with the beta FNA, a mu irreversible antagonist. So that therefore it represents the inactive state of the receptor structure. Here's the side view. This is a beta FNA. It's a very shallow within the binding pocket. This is a top view. The 7 transmembrane domain range as a counterclock orientation. Here's the beta MFA. The one finding of crystal structure is a wide opening binding pocket. And those crystal structure provide used for information for designing drugs, novel drugs, and try to understand the structure base for the biased signaling. This from bottom view. However, in order to stabilize the crystal structure, they have to truncate the N-terminal sequence and the C-terminal sequence. Raise the question regarding the role of those sequences on the overall receptor structure. This crystal structure is co-crystallized with BU72, a mu agonist, and also nanobody 39, which mimic G protein subunit, alpha subunit I, which is shown here, and represent the active state of the receptor. Actually, this crystal structure is identified by the same group. When you compare active and inactive state receptor structure, you can clearly see there are 10 angstrom outward movement from TM6 and a small movement, inward movement, TM5, TM7, and as well as helix 8. Again, so all the N-terminal, C-terminal sequence was truncated. Open question, call those sequence inference the receptor overall, the receptor structure. Here shows the structure of several commonly used mu opiates. Morphine is a benzyl ring structure. Heroin we also call the 3,6 diacetylmorphine. And M6G at the 6th position have guancoronide. M6G actually is 100 potent than morphine, especially in the intracerebral administration. Whereas the fentanyl, here's fentanyl, methadone with a totally different structure, they are all synthetic opiates. Mu opiate actually are complex. And although they are potent in analgesia, they also produce many side effects: respiratory depression, tolerance, dependence, addiction, constipation, itch. And you may already heard a story happening in 2002. A group armed Chechens hold hundreds of hostage in a Moscow theater. To solve the crisis, the Russian special force pump gas into the theater and through the ventilation system. Most of the people passed out immediately and most died. It is believed that the main components that the gas was a fentanyl derivative, leading to respiratory depression, a main cause of the opiate overdose deaths. Actually, I should point out the recent spark of the opiate overdose deaths is really coming from the fentanyl. The holy grail opiate analgesia will be a potent against the multiple type pain. So thermal pain, somatic pain, inflammation pain, neuropathic pain, however, without those side effects. And our goal is to understand the molecular cellular mechanism of the opiate action and try to translate the basic research into developmental better analgesics that maintain potent analgesic action without those side effects. There are several approaches to develop opiate lacking the side effect. One is designed based on the route or site of the administration. For example, a peripheral restricted drug that cannot cross the blood-brain barrier would have no reward, no addiction behavior. Allosteric modulator are targeting allosteric site of opioid receptor, where bivalent or bifunctional ligand are designed based on the heterodimerization of a mu opioid receptor with other GPCR like a delta receptor, NOV, and glutamate receptor, and so on. And the biased ligand are designed based on biased signaling through the opioid receptor, whether it's a G protein bias versus beta-arrestin 2 bias. There are ligands are targeting mu opioid receptor splice variants, which I will talking about today. The concept of multiple opioid receptors has been suggested by clinical observations. Different patients show a diverse response to mu opioids, not only in their analgesic sensitivity, but also in the side effect. If you give morphine, some people feel itch, but some patients doesn't feel itch. Incomplete core tolerance among the opioids leading to the clinical practice of opioid rotation. If a patient, for example, if a patient develops tolerance to morphine, if you switch to fentanyl, another opioid, without a problem. Similar observations were seen in animal models. Different strains of mice have various sensitivity toward mu opioids, incomplete core tolerance among the opioid. And early pharmacological studies use receptor binding assay in vivo analgesia assay with different type of opioids and the opioid peptide to define the three type subtype, mu1, mu2, and M6G receptor. However, owning a single copy OPR1 gene, it is hard to explain all these observations use a single receptor mechanism. Although there are several other possibilities, like a difference in pharmacokinetics or a difference in metabolism, our hypothesis is alternate splicing of the single copy of OPR1 gene create a receptor diversity of complexity, contributing to the individuality of opioid response seen in animals and humans. Driven by this hypothesis, our lab and others have identified and multiple splice variants over past 2 decades. We demonstrate the extensive OPR1 alternate splicing is conserved from rodent to human. And based on the receptor structure, these variants can be categorized into 3 types. The first is the full-length 7-TM C-terminal variant. The next 2 is the truncated 6-TM or single-TM variant. We also show the OPR1 splice variant is region-specific or cell-specific or multi-strain-specific. Many in vitro, in vivo studies show the alternate splicing OPR1 is functional relevant in opioid pharmacology. But more importantly, the splice variants, the OPR1 alternate splice variants can be targeted for developed novel analgesics lacking the side effect. Here I just show the schematic structure of a 3 type variant. This is full-length C-terminal 7-TM variant. They have identical N-terminals, 7 transmembrane domain, identical extracellular loop, intercellular loop, part of intercellular C-terminus, but they only differ in the very tip of the C-terminal sequence due to alternate splicing of downstream of the exons. This splicing is very conserved from mouse, rat, and human. And the function significance of these splice variants has been indicated in mu agonist-induced G protein coupling phosphorylation, internalization, desensitization. Using C-terminal truncation mouse model, we demonstrate different C-terminal tail have important roles in mediate the action of various opioid actions. I will not discuss this study today. Truncate the single TM variant. This only predict a single TM first TM and also conserve from the mouse to human. And although the single TM doesn't bind any opiate, we demonstrate the single TM can function as a molecular chaperone to facilitate the 7-TM expression. So that is have to indicate functional significance of the single TM variant, even it doesn't bind to the opiate. Here is the truncated second TM variant and lacking the first TM, which I'm talking about today, and also conserve from the rodent to the human. We define the function of 6TM variant mainly use the gene targeting mouse models. Here list the 4 animal models. The first model is exon 1 knockout from Pinta's lab. In this knockout, you lost all the 7TM and the single TM, but all the 6TM variant is retained. We made the exon 11 knockout. In exon 11 now, all the 6TM variant lost, where the 7TM and the single TM are retained. We also make a double exon 1, exon 11 knockout. All the splice variants are knockout. No any opioid response in these animals. The Pinta lab also make a triple knockout. In that knockout, we have a mu exon 1 knockout, similar to this one, but also have a delta opioid receptor and the kappa opioid receptor knockout. So in that mouse model, all the opioid receptor are knocked out, only remaining a 6TM variant. So keep in mind, in this model, only 6TM variant is active within the opioid receptor family. IBNTXA is a novel drug derived from naltrexone with modification as a sex position, made by Dr. Majendra, who was a chemistry fellow in Dr. Pasnak's lab. Now he is a faculty in WashU. This is the key compound leading to identify the function of 6TM variant. Actually, by a random chance. By the time Dr. Majendra make this IBNTXA, we also have exon 11 knockout model. So in exon 11 knockout, we knock out all the 6TM variant. When we look at the IBNTX anorexia in the wild type animal is very potent. It's a 10 times potent than morphine in terms of ED50. However, when you knock out all the 6TM in exon 11 knockout, this IBNTXA anorexia is totally lost. Indicate so the IBNTXA anorexia is dependent on the 6TM variant. As I mentioned before, we have a triple knockout showing blue dots here. And when we use the IBNTXA in triple knockout, we label a site similar to the wild type animal. Keep in mind, in the triple knockout, the only receptor remaining is a 6TM variant. So this data suggests the IBNTXA anorexia is independent of traditional 7TM mu, opioid receptor, delta, and the kappa receptor. And we This IBNTXA also potent in other different type of pain models, thermal pain and the inflammation pain, formalin test, complications, fracture ointment test, and neuropathic pain. The IBNTXA can be labeled by IDA-125. We perform the opioid receptor binding assay, use mouse screen. This is saturation studies. We can see in the wild type animal, we label a very high affinity binding site for this site with the blocker, because the IBM-TXA also bind to the traditional 7-TM mu, delta, and kappa. We have to use the blocker like CTAB, Nortrindol, and NorBI to block all other sites, only maintain the 6-TM site. The triple knockout, you can see we label the same site. Again, in triple knockout, the only active receptor is 6-TM. So they label the same site as a wild type with the blocker. At this binding site, we lost in the exon 11 knockout, but it was normal in the triple knockout mouse model. Competition assay. This table just lists the competition assay. Make a long story short. So this binding unique, showing the unique binding assay. The traditional mu, delta, kappa drug and the ROM1, like a NOB drug, receptor agonist, sigma drug, doesn't touch this binding site. It's a KI value over 1 micromolar, like morphine, Tango, DPDP, U50, OFU. However, interesting, this some carbon-like drug like Norbozo, ketocyclazine, and compete this site with a high affinity. It's quite unique, this binding site. To further validate the IBM-TXA analgesia is mediated through the 6-TM variant. We perform a gain function studies, use lentivirus rescue studies, which express lentivirus express MR1G, which is a 6-TM variant. We use this double E1, E11 knockout. As I mentioned before, in that knockout, we lost IBM-TXA analgesia, we lost every analgesia, including morphine, fentanyl, any opiate, mu opiates. Now we give the lentivirus intracerebrally. This just shows the immunostaining with antibody against exon 4, which is encoded by MR1G. We can see this vector control negative, where we label the spinal cord. And then we look at the IBM analgesia, we see time-dependent rescue of IBM-TXA analgesia. By week 3, reach the peak time is maintained 14 weeks after they can maintain 52 weeks. And EGFP, this RNA level, is the lentivirus marker which correlate with the IBM-TXA analgesia. This lentivirus expression MR1G can only rescue IBM-TXA analgesia, but did not rescue other mu opiates like morphine and fentanyl. In mouse, we have 5 6-TM variant. In additional MR1G, we have M, N, K, L. So then we did each lentivirus rescue study for each individual 6-TM variant. This summarized figure showing each individual variant can rescue IBM-TXA analgesia. Here's vector control. There's 5 different individual variants. So all those studies suggest the 6TM variant are essential for the IBM TXA and OGG. We then look at the side effect profile and for the IBM TXA. So we measure the respiratory function, use a pulse oximeter from the Star Life Science. We gave the morphine 10 milligram per kilogram. It's a morphine about the 5 ED50 dose. We see significant induced respiratory depression. However, 5 times ED50 dose of IBM TXA, blue dots here, doesn't cause any respiratory depression similar to saline. Suggests IBM TXA, no respiratory depression, at least in the mouse model. And we also measure the rewarding use of conditional place preference with the 3-chamber apparatus from Med Associate. And you can see the morphine induce a robust CPP, whereas the IBM TXA, no rewarding and no averting. I should have mentioned the averting is the most common for the CABA drug. We also measured the physical dependence of IBM TXA. We gave the drug chronically for 7 to 10 days, then challenge it precipitate with naloxone. Then we look at the withdrawal symptom. This morphine, we mainly use the jump within 15 minutes after immediately after give the naloxone. So we can see the morphine induce significant withdrawal jumping score, whereas IBM TXA have no jumping, very little jumping. So no physical dependence. And we also use the tail merging assay to analyze the hyperalgesia. Here is the morphine, is IBM TXA. And we also look at the constipation. Use a gastrointestinal transit assay with oral administration of charcoal. Here's the morphine, and here's IBM TXA with a different dose. Although compared to saline control, is lower than saline control, but still is more significant than morphine. So we have limited constipation for the IBM TXA. This table just summarizes the results. The IBM TXA compare with morphine. IBM TXA is very potent against the multiple type pain, particularly for inflammation and the pain and the neuropathic pain. And no respiratory depression, no reward, no physical dependence, no strep tail, with limited constipation and the sedation. Therefore, IBM TXA represent a normal opiate with high therapeutic potential. So MP1000 is the second generation of IBM TXA through the SIR screening made by Dr. Magendra. And the MP1000 is more selective than IBM TXA for 6TM variant based on the binding assay. And also MP1000 has similar pharmacological profile to the IBM TXA. And 2 years ago, we were awarded a UT3 grant from the National Institute of Drug Abuse for further develop MP1000 as a part of the grant, which support the startup from Sparring Bioscience. Sparring is a abbreviation of Sparring. This is a Sparring sign here. And for helping, the Sparring is helping for the drug development. So So far we have completed tox studies in rat and dogs, as well as formulation studies, and expect to start a clinical phase 1 trial early next year. We can also classify the drug based on the variant target. As I mentioned before, the IBNKXA MP1000 is 6TM dependent. When you lock out 6TM, this analgesia is lost, where this IBNKXA maintained in the exon 1 knockout. Morphine methadone is 7TM dependent drug. So this also may lost in exon 1 knockout, maintained in the exon 11 knockout. We also have third group, which is both 7TM and 6TM dependent, also based on our knockout study. Those exon 1, exon 11 also partially lost. This contribute to MSG, heroin, buprenorphine, norepinephrine. So to further clarify this third group, we may use the lentivirus rescue studies into in those knockout. We use double knockout, exon 1, E11 knockout, and also E11 knockout. So this I showed before. In this knockout, this buprenorphine and the norepinephrine cannot be rescued by lentivirus expression MR1G. However, when you look at the exon 11 knockout, in this exon 11 knockout, so you lost the buprenorphine analgesia, also lost the norepinephrine analgesia. However, when you introduce the lentivirus, now the buprenorphine and the norepinephrine analgesia can be rescued, maintained. Keep in mind, in that exon 11 knockout, 7TM variant is intact. So that's coming up, the conclusion is, so we consider buprenorphine and norepinephrine are both 7TM, 6TM dependent. 6TM variant also involved other opioid receptor analgesia, like U50 is a kappa agonist. In both exon double knockout, exon 11 knockout, U50 analgesia is a complete loss. When you introduce the lentivirus express MR1G, you can rescue U50 analgesia for both in both knockout. So suggesting 6TM variant involve U50 analgesia. And in DPDP is the delta agonist. And in both double knockout and E11 knockout, DPDP analgesia was lost. However, when you introduce lentivirus MR1G, it doesn't rescue DPDP analgesia. However, similar to buprenorphine, the lentivirus rescue in exon 11 knockout mice, suggesting delta receptor analgesia, the DPDP analgesia involve both 6TM and the 7TM variant of the mu opioid receptor. Clonidine is alpha-2 adrenergic receptor agonist in both double knockout and the E11 knockout. The clonidine analgesia was lost, but however, the lentivirus rescue the chronic analgesia, suggesting the 6TM variant derived from the mu opioid receptor can involve the chronic analgesia. In summary, extensive alternate splicing of mu opioid receptor OPR1 gene produce multiple splice variants, create the receptor diversity, contributing to the complex opioid action. This splice variant can be categorized three major types. and those called exon 1 associate the 7TM variant, or single TM variant, or exon 11 associate the 6TM variant. We also mentioned the mu analgesia can be classified into 3 categories, whether it is 7TM dependent, 6TM dependent, or both 7TM, 6TM dependent. I also talking about the truncated 6TM variant represent a unique target for novel drug with a high analgesia potency and against multiple pain models, but have limited side effect associated with the traditional opiate. I should point out OPR1 alternate splicing is not only limited in the opioid strategy. Based on the initial analysis of GPCR database, you can see 12% of GPCR, 6% GPCR, 7% GPCR have C-terminal variant, truncate, 6TM, single TM variant respectively. Those GPCR exclusive olfactory receptor family. Our study really raised the question whether those alternate splicing variants may expand pharmacological repertoire of GPCR in general. I'd like to thank all the people contribute to this talk, the former lab member and current lab member, and the collaborators, I mentioned Dr. Magenda, and who made those IBM TXA MP1000, jumping the provide their animal models and Jeff model and did some behavior assay. And I should clear, so I'm the co-scientific founder of Sparring Bioscience. In the funding from the NIDA, NIH, and from different funding source. Finally, I'd like to thank Dr. Deborah Pasnek, who is my former postdoc mentor, a longtime collaborator. Unfortunately, she passed away in 2019. He was truly missed. Thank you for your attention. So ready to answer any question.
Thank you very much for your presentation. I'm really, I mean, it's very impressive, the lack of respiratory depression, which you presented. So it's, I would say, critical to translation and development for safe analgesic drugs. Since there are, I will check once again, I can see. And the question, you mentioned that you study the binding or within central nervous system. I have one question. Did you did any research how the developed let's say compounds or candidates for drugs works within entering nervous system or just within gastrointestinal tract?
I try to understand the question. So the question is whether our study
yeah, within and how the compounds or drugs candidates works within entering nervous system, or what are the perspective, or just in effects in gastrointestinal tract.
I mean, how those drugs are working on the central nervous system. So yeah. So actually, we developed this drug. So we have let's see. So this drug we developed, that's MP1000. We actually formulate as IV formulation. So we also working on the oral formulation. We do have currently we do have some candidates for the oral prodrug, which can potentially use orally. So this is an ongoing project. So and also supported by that UHC grant. And so the main thing is we really hoping, although the data are presented here is maybe from the animal studies, the preclinical study. We're really hoping we can get the study in the human study. We can validate our models.
Okay, thank you very much for the answer. So I think that we can welcome our next. Thank you very much once again for your presentation. And I wish good luck with further development and successful implementation, translation into clinical trials, and finally into introduction to the market. That's really impressive. I once again stress that